a major effect on what the receptor is and what in actual biology, and why we may be missing it in a lot of our assays looking at in vitro binding partners at a neutral pH. So PSGL1, I mentioned, is one of the putative receptors, and it's a particularly interesting protein because while this P-selectin is well established to be important for rolling of leukocytes and transitioning through endothelial cells, recently data has been also published to support that this is a negative immune checkpoint. If you knock out this PSGL1 in lymphocytes, the activity of lymphocytes is up. So it's a very interesting candidate for VISTA, given the fact that PSGL1 is negatively charged. So let's see. As I mentioned, not only is the extracellular domain somewhat different from PDL1, but the intracellular domain is much longer. And the tail also includes 2 tyrosines. There's no clear motifs that tell us what the function is, but it is interesting moving forward, looking at different antibodies where we're blocking the function to figure out if the function is blocking on the lymphocyte end or on the myeloid end, or whether it's acting as a ligand or a receptor. So we developed a number of different antibodies. These are just 2 examples of the specificity of the antibodies that we're developing. And we're testing in vivo models with Renca, since it's a very myeloid-rich tumor, as well as a number of different proliferation, activation, phagocytic assays to look both at lymphocyte function and the effect of these antibodies, as well as the myeloid function. Renca has a lot of weaknesses as a model for human kidney cancer, but it is a very immune-infiltrated tumor. You can see on the right by flow cytometry that 16 to 20% of the cells are actually immune cells, and specifically myeloid cells, only about 1% or less, as you heard yesterday. It tends to be a very low lymphocyte-infiltrated tumor, but it's potentially a very good model for these VISTA antibodies because VISTA antibodies have been shown to block tumor growth in myeloid-rich models. And Renca is a very locally aggressive tumor in which, whether it's injected subcutaneously or you can see here, I've injected it into the kidney. Within a month, it grows very quickly, and so it's a very good model for testing therapeutic reagents. So wrapping things up, this is just a subset analysis, not even a subset analysis, it's more just looking at the data that I showed before, that while VISTA is expressed across kidney cancer subtypes, looking at the known or reported receptors, there's different subsets of kidney cancer. For example, here you can see there's a subset that expresses both VSIG3 and VSIG8. This I haven't repeated with the PSGL1 yet because the patent just recently became public, but it tells a story of while VISTA is very highly expressed and is actually more highly in clear cell versus other subtypes, the function of VISTA may vary greatly, whether it's the lymphocyte dependent or the myeloid dependent, depending on the type of tumor it is and whether the receptors are actually in that tumor. So in summary, VISTA is a negative immune checkpoint that's expressed in kidney cancer. Its receptors on lymphocytes is not well established. This interaction appears to be pH dependent, and I'll be testing over the next few years, finishing up this DoD, a number of the VISTA antibodies on these different models to figure out the best antibodies to move forward and to really understand the biology of the receptor ligand in different contexts, because it may be very important for understanding which antibodies to move forward and which patients to test them in. So I have a laundry list of acknowledgement. Gordon Freeman has been an amazing immunology mentor over the past many years as a young investigator and now a not so young investigator. Rupal Bhatt has also been an amazing colleague and now collaborator, as well as Sabina Signoreni, David McDermott, Tony. It's an amazing group we have here of collaborators, and we're doing really amazing things. And I have to thank the Kidney Cancer Association and the advocacy groups, because my early investigator time has really been protected by both the Young Investigator Award supported by KCI and as well as this recent DoD. Thank you.